In this video we will discuss about a new type of measurement to measure the proteins within the cells, that is photoactivatable GFP. So how this will work is that we can use the laser of a specific wavelength to activate our reporter proteins, and then use that after activation use the specific wavelength to excite the proteins and can see the signals whether our protein is present within the sample or not. So this sounds like a confusing statement. We will try to understand that thing through the explanation of this figure. So let us first understand this GFP. So GFP is in normal terms, or its full form is green fluorescence protein. So it will emit green fluorescence. So in the case of wild type cells, what happens is this GFPs exist in two different conditions. First one is the neutral condition and another one is the anionic condition, and this both of them conditions are able to emit fluorescence but at different wavelength. And, and this however, after photoactivation, what has been found is that most of this neutral most of this neutral GFP is converted into the anionic GFP. Therefore, we will see more signals that are emitted from the anionic GFP than from the neutral GFP. And this things has been explained by this panel B. So, in the case of this, without without activation or non-activated GFP, we can see the two signals. First one is around this 400 nanometer emission, and first absorbance is around 400 nanometer, and second minor absorbance is around 475 nanometer, that we can see that. So this is without activation, and after activation we can see that the since most of this neutral has been transferred to the is converted into the anionic, and this conversion is mainly related to this rotation of this T203 or and this decarboxyl, or we can say removal of this E322 decarboxylation. So, and we can see that the anionic the neutral signal got reduced after this activation or this conversion of neutral to anionic, and the more signals we can see from this uh, we can see at this 475 nanometer peak. So what the researchers try to do, or the author did in this paper, is that they try to improve this model to extend that in the normal case we do not see any signal of this minor peak, and when the cells get activated or when these proteins gets activated, we can see these minor signals to a greater extent. And in order to do so, before this paper, some uh, people have reported that if we can mutate this T203 with isoleucine, then we can reduce the signal of minor in the case of minor absorbance in the case of wild type. However, this minor signals is not close to the zero. So the researchers tried different mutation at T203, and they find out that when the T203 is mutated with the histidine, they can see almost nil signal when in the case of wild type cells, wild type GFP, as it is shown here in the panel C. We can see there is no minor peak. However, when the cells get excited, we can see that there is a decrease in the major peak or the peak at this 400 nanometer, and there is a sharp increase in the peak at this 500 nanometer. So there is a slight shift in the wavelength of this minor this anionic peak from this 475 to 500. However, we are able to distinguish whether the cells are when the cells are activated and when the cells are non-activated. So we can reduce the background signal and increase the contrast to a greater extent. And when we compare both the fluorescence, that activated fluorescence of the wild type GFP, and with this mutated GFP, we can see that both the signals give a almost equal contrast, or produce a almost equal fluorescence, or in other terms the normalized fluorescence are almost equal. However, there is just a slight shift of this mutated GFP uh, towards this 520, whereas this wild type GFP fluorescence activated wild type GFP fluorescence is around 500 or 475 in this figure. So so far in this panel, this figure shows that they are able to produce a mutant GFP that can provide more contrast between uh, between photoactivable GFP and non-photoactivable GFP. If you need to understand more about this photoactivable GFP, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.